So thank you very much, Rohini, and thanks for the invite to give this talk. So today I'll be talking about how we can harness African genomic diversity for global precision medicine. Like I've been introduced, I'm Segun Fatumo, a professor and chair of genomic diversity here at Kumere. I'm also the head of NCD Genomics at MRC in Uganda. I'm going to start in case if anybody don't know about FURI. FURI is a Precision Healthcare University Research Institute. Yeah, and our work is focusing mainly on prediction, precision, and prevention. We are doing that by integrating analysis of large-scale data sets using different kind of omics data sets across looking at the different ancestry in East London and globally. So at FURI, currently we have 3 full-time professors. So that include Claudia, who is our director, and Mark Brantley, who is also the chair of and also center lead for Therapeutic Innovation Center. So Mark also is the new deputy vice principal for research and innovation. And myself, we are full-time. We also have a part-time professor, including Mark, Simon, and Paul Frank. So if you have any question about FURI, please just can also ask me that. So today I'll be talking about, my talk I've been put into 3 different parts. So the first part, I will talk briefly about genomic diversity and its implication for precision medicine. And I'll move talking specifically about our work on genomic discovery and genetic risk score using an example from African population. So now we end by talking about the approach to increase genomic diversity and achieve much more accurate genetic risk prediction across all populations. So like you may know, there has been an evolution of medicine. So in the past, medicine was practiced according to signs and symptoms that are presented by the patient, and the physician or the doctor is always the person with the know-how all of how to take care of the patient using their own intuition medicine. So currently, so medicine is based on evidence, and of course, we see a lot of that during COVID. We had a lot about a clinical trial during COVID. So now and in the future, so medicine will be practiced according to an algorithm, and this algorithm will take into consideration the patient characteristics, and that include their genomics, their epigenetics, their lifestyle. This is what constitutes what I'm defining as precision medicine. So here's an example. On the left here, we see an example of a patient population, which shows that there is diversity in patient population, and this diversity can be their ancestry or their ethnic group. And on the right here, you see an example of treatments. Let's say this is a treatment A that is being applied to the whole patient population. This is like the standard approach where this treatment is applied is only effective in about 20% of the population and become a waste in the remaining people in the population. So a much more tailored approach is an approach where people are classified into different groups, and an appropriate treatment is given to people according to their characteristics, as I've defined in the previous slide. So if I give an example of PCSK inhibitor, from this inhibitor, there have been discovery of a highly effective new class of drug that very, very effective in lowering bad cholesterol and also helping to prevent heart attack. So this inhibitor was discovered because some African ancestry individuals were included in a study. So in that study, they found that those African ancestry individuals have some genetic variant within the PCSK9 gene that is helping them to lower cholesterol naturally. So the point I want to bring out here is because is that we have today a treatment that is effective for African and non-African because African are included in that particular study. So there was a lot of inclusion in that particular study. So the point is here is that we are doing ourselves harms if a genomic study is not diversified to include all populations. including all underrepresented population. Like you might know, Africa is the most diverse continent in the world. It's both genetically diverse, culturally diverse, in terms of ethnics also diverse, in terms of linguistic also very, very diverse. Give an example, Africa has about 3,000 different ethnic groups, which is really clustered also towards their genomics, and they are speaking about 2,100 different languages. So African is diverse because there have been so many multiple genetic studies that confirm what is known as the African out of African theory. So about 60,000 to 100,000 years ago, so the modern woman migrated out of Africa and they started to populate the globe. So when they left Africa, so they carry with them only a small part of the African genetic variation with them. So this makes African the most genetically diverse population in the world. So the emphasis I want to make also, if you look on the right here, on the left here, you see a map of Africa, which shows that there is even larger genetic differences within Africa than between Africa and other population, or than between African and say European population, or between Africa and Asian population. So there might be even that larger genetic differences between say South African compared to Uganda than between Africa as a continent with European population. And here is an example from one of the work from our colleagues at the H3 Africa, where samples were collected all across different countries in Africa. And that also includes sample collected within from Uganda population. I'll give an example here. So sample was collected in a site here called UNS, another site here called UBS. And if you are familiar with the principal components analysis, PCA, on the right here, so you see the sample that was collected in Uganda here is now is tailoring towards Central Africa, and another one in the same country tailoring towards East Africa. So you can see a big diversity just within one country, just in Uganda, in the same country, but the diversity was really much more larger. So this diversity has implication. So number one, it can help us to understand the human evolution. I can also give an implication of that genetic study in non-African population, including their clinical relevance, may not be generalizable to African population. I mean, study from a European population or Asian population may not be generalizable to African population. It might also mean that there are many genetic variants that are yet to be discovered if we don't study all population. And the last one here is that, like I mentioned previously, so findings from one region within African population may not actually be transferable to another region within African population. And I'll give an example from our study. So there's a drastic lack of diversity in genomics, despite the record. Here, I show some work that done in the last few years. In 2016, you see most of the genomic study have been focusing primarily on people of European ancestry. Here is showing data from 81% of European ancestry and only 3% from African ancestry. If you move down the line, 2019, you even see a further decline in study from African ancestry from 3% to 2%, and further decline to 1% in 2021. So you see, we're not really making a lot of progress in terms of genomics that have been done in non-European ancestry individuals. So the 1.1% of African ancestry that I mentioned that have been done is mainly actually African in diaspora. I mean, African in the UK Biobank, or African in all of us in the US, or African in the Medium Veteran Program in the US. So if we continue to study a small proportion of African in diaspora, and we just group them together and we say African ancestry, we continue to broaden genomic imbalance and fail to capture the genetic diversity within African population. And yeah, I made that emphasis in this paper that we published in Nature, where we emphasize that African Americans or African in the UK don't really capture the African genetic diversity. Just remember that I mentioned Africa is home to about 3,000 different ethnic groups. And I give an example from study that collected sample from about 5,000 people. And looking at those sample individually, you see that most of those samples are coming from just one region or one country from the continental African population. And here is just to talk about the UK Biobank, which many people in the room today may be familiar with. UK Biobank has about 500,000 individuals, and that includes about 38,000 individuals of non-European ancestry. But if you look at in the publication so far, you see that about 93% of publication excluded non-European ancestry, including Africa. So again, this is showing that analysis has been done, progress has been made in terms of genomic medicine and precision medicine, primarily on European ancestry, and non-European ancestry population has been neglected. Okay, so I'll move to the second part, which is about what we've done so far. Here, this is work that from the Uganda and Genome Resource in 2019, where this paper was published in Cell. This was reported as the largest ever genomic study of Africa. In this study, we genotyped about 5,000 individuals and also did whole genome sequencing on additional 2,000 individuals. And we had extra data from a colleague in Africa, from South Africa, by about 1,000, and also another cohort called Adam, about 5,000. Altogether, 14,000 individuals, considered largest ever genomic study in Africa when it was published. And if you compare this with a study from like a genetic study of height that was published about 2 years ago, that's about 5.4 million people, you see that not a lot has been done within the African population. So in that study, of course, we see evidence of population substructure. This is just a data from 2,000 people from Uganda. You see kind of confounding, and that have been need for like some specialized approach, like mixed model approach to analyze this data. And we also identify 9.5 million novel variants in just data about 2,000 whole genome sequencing. So those novel variants were not present at that time in the 1,000 genome project, phase 3, the African Genome Variation Project, and also the UK 10K reference panel. It's 9.5 million is a lot of novel variants in this small data set. We also did a genome-wide study, GWAS, of different cardiometabolic traits, about 34 cardiometabolic traits. And this table just to show where we found novel and distinct association signal in our GWAS. But what I want to emphasize here is the last two columns here, which is the minor allelic frequency in African population compared to the allelic frequency in European ancestry. You see that most of these variants that were found to be novel associated with the cardiometabolic trait are actually monomorphic in European population and maybe mostly common in African population. So this means that even with the largest ever genomic study in European population, this variant would not be found because they're actually absent in European ancestry data sets. So we also assess the heritability for all those 34 cardiometabolic traits. So we found that height, as an example, is less genetically determined in Uganda relative to what is known in European ancestry. We reported the narrow sense heritability estimate for height was 49% in Uganda compared to about 80% in European ancestry. And in contrast to that, we found the heritability estimate for LDL was significantly much higher in Uganda, about 54% compared to about 20% in European ancestry. And here is another study where we leverage multi-trait lipid traits and did a multi-trait file mapping in about 105,000 individual of African ancestry, both African ancestry in continental Africa and also in diaspora. And we identify several novel loci that would not have been identified, even with the largest ever GWAS of European ancestry. Again, this is emphasized the need why we need to study all populations. And here we're trying to do genetic prediction for type 2 diabetes in continental Africa. So on the left here, so we developed PRS using multi-ancestry, about 1.4 million people. Developed other PRS using African ancestry individuals, just about 56,000 people, and European ancestry about 1.1 million people. And we tested this PRS in the continental African data set in South Africa, and did a validation in Adam cohort that has individuals from Kenya, Nigeria, and also from Ghana. So what we found is that for using the best PRS, we found that the PRS that we developed from the African ancestry was actually performing much better than from the PRS from the very large data set from European ancestry and the multi-ancestry PRS. And also looking at the validation data sets, we saw that the PRS was doing much better also from the one that was developed from African ancestry. From the validation data, we have R-square of 2.92 compared to 0.13 from European ancestry. So again, this is showing that because we have some data set from African ancestry, we can develop PRS that is much more predictive using that instead of using the PRS from the multi-ancestry or from European ancestry. From our PRS study, we saw that the participants on the 10th decile of the African-derived PRS, they had more than 3-fold higher risk of developing type 2 diabetes risks compared to those on the first decile. And we also saw that on average, those on the 10th decile, they were diagnosed for type 2 diabetes about 2.6 years earlier than those on the first decile. And we also took the best PRS. We checked them by different country. We compared that with the PRS that developed from the multi-ancestry. This was important because at that time, what was in the literature was that the multi-ancestry PRS was just as good for all population. And here we show that the multi-ancestry PRS was not actually as good in all population. Of course, it was doing very well in the Nigerian population and Ghana population, which are West African population, but not as much in the Kenya population, which is the East African population. And then we show the discriminatory ability of PRS. If you model the conventional risk factor like age and BMI, we find the area under the curve to be 67.9%. So we compare that if you introduce genetics, which is PRS, into the model, we show that the AUC was 69.8%. This show an improved discriminatory ability of 1.9%. So we did the same thing for lipid traits. What you will see is on the plot here is the African-derived PRS were doing much more better compared to the multi-ancestry and European ancestry. Very similar to what you see from the European ancestry, from the type 2 diabetes PRS that I showed in the previous slide. So because we were very excited about the PRS producing better from the African ancestry to South African population in Africa, we also tested this in another population in Africa here, comparing the PRS that was doing very well in Zulu population, South Africa, and then check that also in the Uganda population. We see that even though the PRS is doing well in one particular region in Africa, it doesn't mean that it will do well in another region within the same African population. Again, this takes me back to what I was saying at the beginning, that there is even larger genetic differences within African population than between African population and other populations. So this work was published in Nature Medicine. So recently, this is our new work that was just published a few weeks ago, last month. Here we show the first major proteomic study in continental Africa. We're trying to address the severe underrepresentation of African in omics, not only in genomics, also in proteomics. So here we analyze about 3,000 protein from about only 500 Uganda individuals. We put them into two categories. 103 of them with diabetes or pre-diabetes, and the remaining 362 of them as control. And we combine the proteomics and genomics data to map the protein constitutive trait loci PQTR. So what we found, the first one was that we investigated the association between the protein level and the cardiometabolic traits that are measured in the Ugandan genome resource. Here we found a total of 288 protein that were associated with HB1C, 142 with HGL, and 46 with LDL at FDR of 5%. And we also found an association of, such as the ARCC1, which we found to be associated with HB1C, and HGL has been known previously to affect glucose intolerance in an animal model, causing an auto-inflammatory response that leads to fat loss and insulin resistance. So in this particular part of the work, so we sought to identify differentially expressed protein level between the cases and the control. Here we identify 88 differentially expressed protein. 54 of them were significantly upregulated, and 31 of them of the protein were downregulated. And looking at the PQTL, we identified 399 independent PQTL association, 340 of them was cis PQTL, and 53 of them was trans PQTL. Just to be very a bit more specific, 37 of them had not been previously identified before in the population. Just take into consideration that this will just say a moderate data set of about 500 individuals from Uganda. 58 of those PQTL were African ancestry specific PQTL that had not been previously documented before. So we also perform a co-localization analysis in order to determine shared risk variance between the PQTL and type 2 diabetes using large-scale multi-ancestry GWAS. So one significant co-localized signal was identified with a posterior probability of about 96%, which indicates very, very strong evidence of a shared causal variance. And so the variant is RS6075339, identified as a PQTL regulating SIRP alpha expression. And I will show a bit more of this in the next slide. So for this particular protein, so we found a previous evidence of this to be previously associated with type 1 diabetics. Okay, just to give you a wrap up of that new study. Of course, I didn't show much about the AC show causal association in terms of the Mendelian analysis that we did between type 2 diabetes and AC inhibitor. So, like AC inhibitors is already known to be clinically used for hypertension. And what we saw from the MR analysis that we did was that this also may reduce the type 2 diabetes in the incidence in high-risk individuals. And this also gives a room for personalized medicine and novel drug targets. So I'm going to now move to some of the previous studies that we did in 2019, 2020. So we did this study looking at kidney disease in African ancestry. This was also a moderate data set, about 3,000 individuals. So we were trying to find association between kidney function and genetics. So we identified and validated two kidney function loci, one at a GATM locus, and the second one at the HBB locus. If you're familiar with HBB locus, it's a locus that is known for sickle cell disease. So since then, we try to expand on that study. We'll be working on the multi-cohort study of kidney function in African population. So here we were aggregating data sets all across different sites in Africa and trying to see how best to pull this data set together in terms of analysis, taking to consideration the kind of diversity within different regions in Africa. So what we decided to do was to run GWAS according to different regions. So we run GWAS on East African population, put data set from all those from East Africa separately, Western Africa separately, and Southern Africa separately. And we did a second stage GWAS where we now pull all the regions together. And the last stage GWAS where we put the region within Africa and also African and diaspora together. So what we found is very surprising. So we found that when you run GWAS of kidney function according to region, so we didn't find any association that is common among those regions. Again, this is showing how the genetic architecture within those regions, particularly in this case with kidney function, might actually be different. So here, if you look at the novellos that we found and check the effect allele frequency from African population compared to European population or East Asia population, we see that all these variants, this local that we found to be novel and associated with kidney function in African population, they are mostly monomorphic in other population, including European and East Asia population, or very rare in those populations. Again, this is emphasizing why we need to analyze all populations. So in the second stage of the analysis, where we now pull all those regions together and in GWAS, of course, you find the very strong signal in African population, which is a signal as a GATM signal, but you also observe attenuation. What that means is that all those signals that you previously see, if you analyze per region, they just disappear when you put them together. So you see an attenuation of the regional kidney function signal. So the last stage, yeah, we put together on the top here is the Mata plot of the continent Africa GWAS, and here is the Pan African one. Of course, because the Pan African one much more larger, you have a new signal that you will not see with the continent Africa. So here we're able to present 20 independent loci and 4 novel association looking at the Pan African GWAS meta-analysis. So we're also very interested in looking at the genetic prediction of kidney function in African population. So we developed the PRS using different data sets. We look at data set from the South Africa separately, West Africa, East Africa. Look at the one from the continent Africa, the one in diaspora, and also Pan Africa. Of course, the one from the Pan Africa, they're much more larger, over 100,000, and the one from the region are much more smaller data sets where the PRS were developed. So we picked those PRS and we now tested those PRS on the Malawi dataset and also had the validation dataset on the Malawi dataset. Of course, you expect that the bigger PRS would do much better in Malawi dataset, but let's have a look at what actually happened. So we saw here that the predictivity and the transferability of genetic risk score may actually be driven by genetic distance, because we found that the PRS that was developed in the South African population was doing much more better in Malawi population compared to other population. So the PRS I show here from South Africa, just not as big, about 5,000 individuals, doing much more better than even the PRS that were developed from the Pan-African one or from the diaspora population. So this is showing that the PRS might actually be the transferability or prediction might be driven by genetic distance, because the PRS from the South African population is the closest to the Malawi population, which is also another South African population. Of course, with the fine mapping, we're able to establish the causal missense variants like the RS334 in HBB gene that is known for sickle cell, and also other variants. So we found some of those variants having the posterior probability of 1% and just one variant within the credible set. So we also were trying to look at the role of APOL1 haplotype in low kidney function continent Africa. If you're familiar in this domain, I mean the genetic of kidney function, we know that the APOL1 gene is known to be strongly associated with kidney function in African ancestry. But this was actually in the African American ancestry. And now, this has not been tested within the continent Africa. So what we did here is to look at those APOL1 variants. They are called G1, G2 variants, and see how they are associated within our continent Africa population. So we see that this variant, the the increased risk of developing kidney disease was approximately about 13% in African ancestry variants for those that carry those G1, G2 variant. So we checked this. So we observed that in our population, the overall frequency of the APOE1 high risk is significantly lower, about 5%, compared to 13% that you see in the African American population. But we also saw that the frequency is not the same all across African population. If you look at the South African population, you see the frequency about 8.6%, and look at the East African population, like Uganda, you see the frequency only about 1.38%. So you see diverse frequency of the APOL1 across different populations within Africa. We also found a very limited association of APOL1 with a EGFR kidney function compared to what you see in African ancestry. So again, emphasizing the need why we need to study our population and look at the diversity within Africa. And so this work was recently accepted in Nature Communication and is currently in press. So now on the right here, I show the map of Africa and I'm showing where data are currently available. So there's now a growing number of African genome resources that they are capturing that diversity that I mentioned. This would be a great value for discovery and also for genetic prediction that can be much more generalizable broadly across a global population. Remember, I mentioned previously that there's more diversity within African population, which will be very helpful for discovery and also for genetic risk prediction. So in order to achieve the accurate genetic risk prediction and transferability across all population, so large-scale African data sets are necessary for PRS to achieve its potential. So as I conclude, I'll just talk about our approach to increase diversity and achieve accurate genetic risk prediction across our population. So our group here at Kwemere and also in Uganda, trying to focus primarily on generally NCD genomics, looking specifically on kidney disease, looking at cardiometabolic traits, looking at psychiatric genetics. So we are doing this by leveraging existing data sets within continental Africa and also in diaspora in African American and African in the UK. And we also are working on building our resources. In the last few years, we've been building the 10K Uganda biobank, building a biobank that will be much more bigger than currently what we have. So we also have a vision of working on what we call the 100K Black British biobank and do a phenotyping. Very important, we've been working the last few years on just genomics. We're not going to be transiting to do other omics, try generate omics data set across the diverse population globally, and also generating omic data sets, very specific to African population. So recently, we were funded by the MRC to establish Kidney Gen Africa, in order to facilitate multi-center research in Africa, to enhance large-scale genetic study, and also deliver both research and training excellence in genomics of kidney function in Africa. So this new consortium is led by myself and my colleague, who is based in South Africa. We have people representing different countries in South Africa, in Malawi, in Uganda, in Nigeria, and also in the US. So just to emphasize what we do in terms of Kidney Gen Africa, and if you are interested in what this partnership is trying to deliver, you can have a look at the website called kidneygenafrica.org. So as part of this, because we know, I showed previously one of the slides where data is available, like in the UK Biobank, so data are not even being analyzed. So one of the reasons for that is because capacity to analyze this data set is very limited, and very limited within people that are underrepresented population in science generally. So one of the things we're doing in Kidney Gen Africa is to build capacity, train people of African ancestry or African heritage, both those in the UK or in the West and those in African population. So we're doing that by doing workshops. We're going to be doing specialized workshop over the next 4 years, training about 120 Africans of early career, people of African heritage of or the early career researcher. There'll also be a lot of exchange, such exchange. So this one is locally looking at people that are based in Africa. They come to the UK or come to the US with our partner and get some training for about up to 6 months and go back to their country. We also have some funding for like bursary, giving about £20,000 as a seed funding for people of African heritage to start a research looking at genetics of kidney function. We've now employed 2 postdocs. Very important reason why we need to do the training is because we also need to build the soft skills, skills like science communication, leading consulting, and grant writing. So yeah, I'm just also give an example of initiative we're involved in trying to build a biobank in Africa. So just to also mention here is that we just recently completed this biobank that we're building in Africa. We recruited about 10,141 participants in Uganda. We have sampled and stored serum, plasma, whole blood, urine, and DNA. And we plan to keep on expanding on this biobank that has been built. So as I conclude, I'd like to mention that we know in the UK currently there are about 2.5 million people of black ethnicity that live in the UK. So that represents about 4% of the entire population. 1.5 million of those people are black Africans, and 1 million people are black Caribbean. Like you might know, disease such as hypertension, type 2 diabetes, and other cardiovascular disease are much more prevalent in this population. And so the idea of a Black British Biobank is to have a much more comprehensive focus on the people of Black British people to be able to understand why we have higher prevalence of hypertension, type 2 diabetes, or cardiovascular disease in this population. So lastly is the omics. We're currently working with a big biotech company, a big biopharmaceutical company to generate omics data sets. First of all, looking at the people of African ancestry and also much more globally. So in conclusion, genomics is a powerful tool for advancing precision medicine, but only if it includes all populations. And I give me say again and again that Africa has the greatest genetic diversity on Earth, but this particular population remains severely underrepresented in genomic studies. Our work from just a moderate data set that I've shown today, I'll be able to demonstrate discovery of novel genetic loci that are unique to this population. I also showed improved project risk prediction and file mapping in African population. So we now must add to generate large-scale data set, and this is one of the things that we are doing, both within continental Africa and also planned for the future in the UK. Generate large-scale African genomic data set that include Afrocentric biobank. We must build capacity, locally and infrastructure for research and training, and foster equitable collaboration and leadership within Africa. So we must add to diversify data and democratize discovery of global precision medicine. So I would like to end by just acknowledging members of my group who are based here in the UK and also in Uganda who've done all the work I've shown today. Also, I'd like to acknowledge the funding agencies that fund our work, including Welcome, MRC, and also NIH. So thank you very much for listening. Very happy to take any question.